Hello and welcome to Pharma Television News Review here at Biopharm America in Boston. On this show, I have two guests. I have Dr. Alan Ezekowitz, who is a Senior Vice President at Merck. I'll come back to you, Alan, in a moment. And Professor Laurie Glimcher, who is a Professor of Immunology at Harvard Medical School. Welcome to the show, both of you. Alan, you're responsible for a range of therapeutic areas at Merck Research Laboratories, but we're here to talk about the bone research collaboration between yourself and the Merck and with Professor Glimcher here at Harvard Medical School. And I want to talk about that research, Laurie, and your discovery, because your discovery was somewhat of a surprise to you. Could you just tell us, well, part of your discovery is a surprise to you. Could you describe how that happened and where it's placed you out of immunology and into the area of bone research?
Sure, be happy to. We are actually primarily an immunology laboratory, and we were looking for a novel transcription factor. That's a regulatory protein that controls gene expression in the immune system. Yep. We isolated an interesting candidate, and to figure out what it really did by knocking it out in mice. So you understand it's sort of the gold standard for figuring out what any given gene does. Yep. To our disappointment, there was no interesting phenotype in the immune system. It looked pretty normal. But we did note that it was very difficult to extract bone marrow from these mice. And I remember the day that my graduate student and my postdoc came into my office and said, you know, there's really no immune system phenotype here. And I asked them, well, have you looked at every single organ? Did you look at the bone marrow? And they said, well, you know, that's the problem, is that we can't really extract cells from the bone marrow. It seems to be occluded. And I said, well, why don't you go down the street to my father's laboratory, who's a bona fide skeletal biologist, and take an x-ray of the mice. And lo and behold, the mice had very high bone mass, but the bones looked normal in shape. So we did a number of experiments over the next year to figure out the mechanism and to figure out more about it, and we found that the function of this protein, which we call SNRI3, mapped to the osteoblast, which is the bone-forming cell. So its deletion increases bone formation. It activates the osteoblast to make more bone synthetic proteins, and it is a process that increases over time so that the older the mice get, the more bone they have. Right. So this immediately appeared to us to be an exciting target for osteoporosis.
Sure. So it's not a transcription factor, it's an adapter protein?
It was thought to be a transcription factor, but in all the experiments that we've done, we have seen no evidence that it has the classic characteristics of a transcription factor, which is to bind to DNA regulatory elements. Sure. So we think it is actually more of an adapter protein, and it probably has a number of different substrate targets in the osteoblast, which may be one reason why it's such a powerful regulator of osteoblast activation and differentiation.
Right. So the collaboration with Merck is to better understand this pathway, and in so doing, to discover new targets potentially for osteoporosis. Is that correct?
I think that's the overall goal.
Right.
One possibility is that this protein itself would be a very good direct target, and sure, we have recently obtained some evidence suggesting that that is the case. The other way to go, the other avenue to explore, and we've done this extensively with Merck, has been to understand the whole signaling systems that stem from this factor in the osteoblast by doing extensive genetic profiling, protein profiling, phosphoproteomics, making additional genetic mutant mice to lay out the whole pathway of signal transduction in the osteoblast that is governed by this important factor.
So Laurie, for you and your research, how important was the Merck collaboration? I mean, clearly there's several aspects there. Obviously, there's hopefully some money component to it, but does it allow you to get access to further research from the Merck organization?
You know, for us, it was absolutely critical. There is no way that we could have made the progress we've made over the last 2.5 years without Merck as our partner. And Merck is really a true partner. This is not a situation where Merck writes a check and says, good luck. This is day-to-day communication between the scientists at Merck and the scientists in my laboratory, literally everyday communication by email, by phone, with monthly meetings face-to-face to go over milestones, accomplishments, where we need to go, what else needs to be done. We've had a wonderful project manager at Merck who has really put his heart and soul into this, and it's been incredibly productive for both of us.
So Alan, you know, pharmaceutical companies have lots of collaborations with university professors. Some of them are successful, some of them are not successful. From your perspective, from Merck's perspective, why has this collaboration worked well? What is it that makes it different besides, obviously, Professor Glimcher here being an excellent scientist?
Right. I think having come from academia, I understood what the strengths are in terms of what is done extremely well in academia, and having experienced what the pharmaceutical industry can do. The old model would have been that Merck scientists would have read the paper and then separately try to recapitulate the results, built all the genetic models, and sort of worked on a parallel path. And what we decided to do was, why would we want to do that? Why don't we just go to the people who are the experts and provide them with the expertise of what they know how to do best, which is to understand the innovation and how the pathway works. And we'll provide them with the tools that they need to actually translate that as discoveries, as well as for us to make progress in terms of the way we're going to address things. And I think that there were skeptics within Merck when we entered into this collaboration, just like there were skeptics within academia. And I think that the model and the paradigm that I represented and understood was, first of all, we protect the academic freedom of the people working in Laurie's lab. The postdocs and the graduate students are free at any time to talk about the information that they produce, to publish it in peer-reviewed journals, and so that this is a good attempt to actually further their career. And the other important partner in this was Harvard, who actually understood that it was very important for the university to protect its mission, but also that Merck had a mission too, which is to make drugs, and that part of that mission includes protecting the intellectual property around some of the small molecules that we develop. And obviously that's also in the interests of the university, because there's a royalty stream that goes with successful inventions if they reduce to practice.
So how does the science actually work? Is the science actually done at Harvard, or is it done at Merck laboratories? Where?
It's done at both places, which is really one of the, I think, key components of this. That we do 100% of research in our laboratories, and then Merck uses its fantastic resources, which we really wouldn't have access to otherwise, to do more research. This is a complete partnership. And you know, in selecting our partner, I mean, we were approached by a number of different companies who were interested in it. You know, it became obvious to us very quickly that Merck was the right partner to choose, because they know how to carry out collaborations with academia where you harvest the best from both.
Sure.
So we're not doing high throughput drug screening in our laboratory. That's what Merck really does well. What we do is help them think of new screens and help set up such assays, and then go back once we have a smaller list of interesting lead candidates and test them in our secondary and tertiary assays of bone development.
Right. So where are you now on the collaboration? You clearly have moved down the path quite far. You're actually doing some form of drug assessment, but importantly, as I understand it, you're still at the stage of target identification, target validation. Is that correct?
Several screens have been carried out. There are some compounds of interest that we are looking at, and there are some novel targets that stem from the signaling pathways whose phenotype we are also examining. I would say we have several lead candidates.
From the research? From
the research that we are actively pursuing.
And Alan, back to you. I mean, you've described this as a paradigm. I mean, is this for a company like Merck? Is this a reproducible type of collaboration? Can you escalate this or develop it for other collaborations, or is it always going to be down to to individual cases?
Well, I think what we learned from this is obviously Laurie is a very special scientist, and she has a very keen understanding of how you translate science. And so what I think we take home from this is, yes, this is a paradigm, but just like Laurie had to choose the right partner, we also have to be cognizant that we have to choose the right academic partner to work with, so that all the incentives are really aligned and the plans are aligned. And we've certainly learned how to do that. And yes, we have replicated it in other disease areas, which I oversee, and also other disease areas within the company. So we have a similar collaboration in the respiratory area, two of them actually, along these lines. From my perspective, there's a limit to how many of these things you can do because you need to manage them very carefully, and so that it's important to focus and to be selective.
And I thank you both for coming on the show and telling me all about the collaboration. Thank you very much indeed.
You're welcome.
It's a pleasure.